The overall goal of this procedure is to generate and select stable transgenic lines of the novel model organism Ostreococcus tauri, a marine picoeukaryotic algal species. This is accomplished by first generating cell pellets from 50 ml of dense algal culture per transformation. The second step is to mix the cells with linearized DNA and then to perform the electroporation. After diluting the transformed cells in fresh medium and incubating overnight, add 0.2% low melting point agarose and a selective agent and pour the mixture into small petri dishes. The final step is to pick resulting colonies that will have grown after 2 to 3 weeks incubation in constant blue light. Ultimately, selection in liquid medium and subsequent molecular analyses such as PCR and southern blot are used to show successful integration of the construct into the genome of the generated transgenic lines.
The main advantage of using this organism over existing model organisms like Gerben is the greatly reduced genomic complexity that allows the study of regulatory networks in a background with virtually no genetic redundancy.
Although in my lab we've used Ostreococcus to study the plant circadian clock, it can be used to study many other complex cellular networks, so the implications of this technique extend well beyond the realm of plant biology.
Visual demonstration of this method is critical because although the individual steps appear rather simple, proper handling of the cells and an efficient workflow are essential for obtaining a good result.
Parts of this procedure will be demonstrated by Katalin, a research assistant from our laboratory.
Ostreococcus cells are cultured in artificial seawater, or ASW.